Alrighty, sounds good. Well, good morning, everyone. Couple of little announcements here to begin. I've got quiz 6 from chapter 6 is up. I wanted to wait till after the test to put that up. Didn't want to put too much on everybody, but I'm still missing a lot of the labs from the little soils lab that we did. And again, even it's late, but still turn it in because some credit is better than no credit. Also, we still have a couple of people that have not taken the test, so that's not good. So I've opened up the test for another day or two. So we can discuss it on Wednesday. Really can't go beyond that because it's time to get through this unit. Anyway, yeah, do the test. And we did have a couple of technical difficulties. And, if you have a technical difficulty, best thing to do is just to let me know that you had that as soon as possible. If without those technical difficulties, we would not have kept it open any longer. But I still have like 3 people that need to take the test. So, get that in. It's a pretty major part of the grade. So anyway, let's move on. This is getting into the second part here of chapter 7. Again, we're working on plant growth and development. And we started kind of getting into the sexual reproduction of plants. And we're going to kind of finish that today. And then I'll probably wait until Wednesday to put up the quiz for chapter 7. Just to space it out a little bit and give everybody some chances to catch up. One final announcement there is that very few people have signed up for their crop for their project. So unless you guys are just having difficulty finding that, if you are, again, let me know. Otherwise, please get onto the discussion board and sign up for your crop for your project so I can give you guys more details and pair you up for teams for the project. All right, well, let's get into chapter 7, part 2. And let me share my screen with you. All right. So again, plant growth and development, part 2, getting into the sexual reproduction. And, most of our crops, again, we're dealing with angiosperm crops, the flowering plants. So it's very important to know about the flowers and how pollination occurs. So I edited the PowerPoint quite a bit. I think a little bit of the information is just little vague, so some of that I've kind of improved on a little bit. First, this is kind of your typical flower. Of course, as we know, flowers, there's today over 300,000 named species of plants. So with that, there are an extreme variety in plant flowers, their shapes, their sizes, their colors. And really one of the biggest things with plant flowers is that they all still contain the sexual parts, the sexual organs of the plants. And again, know these flower parts, they change from plant to plant. For instance, like we were looking at a dogwood flower, you would assume that the large white showy part of the flower is the petals. That's not actually true. In that case, it's the leaf bracts. So these flower parts change a lot, and that's why in botany, the taxonomists have lumped plants together according to their groups by their sexual reproductive parts. So the big thing I want to point out here, of course, we have the female part of the flower. And really that's the whole thing. We've got the stigma, which is right here on the very top. That is the kind of sticky receptive part of the female flower. And then at the base down here, you can see the ovules inside what are the ovary. And we also have the style, which is kind of the tube there that the pollen ends up traveling down, or tubes inside of that anyway. The male parts of the flower are the anthers, which we have that kind of the pollen containing part is the anther there on the very top, and then the filament is the tube that transfers pollen. And most of the angiosperms have kind of co-evolved with other animals, whether it's insects or mammals or birds, whatever, in part by the flowers, in part by the fruits. So in the flowers, we have this kind of nectar area that is outside of the ovules and ovaries, and that's what attracts many insects and birds to the flowers, and that causes cross-pollination. So know the parts of the flower, at least the simple parts, and of course the male gametophyte, kind of the flower's version of the sperm, are the pollen grains. You don't call it pollination for nothing. And again, pollen grains, of course, come in many different shapes and sizes, and some of those, often because of their shapes and sizes, do cause allergic reactions to people. Don't blame the female flower parts, that's the males. It's their fault if you have allergies. So what happens when we have pollination? Basically, that's the transfer of the pollen from the anther to the stigma, the receptive part of the female flower in the flowering plants. It's a little bit different in the gymnosperms and some of the simpler plants. Of course, most of the simpler plants, which we might use in kind of ornamental horticulture, they're not really grown as crops in agriculture, but they can be useful. They often grow by spores, or always grow by spores. Mosses, the club mosses, ferns, etc. We move up to the gymnosperms, that's when we get into cones. And then finally in the angiosperms, we actually get fruit. So basically, we have two types of pollination. We've either got self-pollination or cross-pollination. And that can occur in the same flower, which is self-pollination, or different flowers on the same plant, still self-pollination. Different flowers on different plants, but still the same genetics, that's also self-pollination. And then we have different plants with different flowers, that's cross-pollination. And of course, that pollination depends on the biology and the evolution or co-evolution of plants with other organisms. We're really looking at the structure of the floral parts of the plants, and interestingly enough, genetic diversity seems to be correlated there. And what I mean by that is that if the plants need a wider gene pool in order to survive, then they tend to be cross-pollinating. Whereas if they don't seem to exhibit the effects of inbreeding depression, for instance, then you have self-pollinating plants. A self-fertile or self-pollinating plant produces its fruit and seed with its own pollen, of course, without pollen from another plant. Then we have self-sterile plants, which are cross-pollinating, and usually that's because they need kind of that genetic crossover. They cannot set fruit and seed without their own pollen, but require pollen from another plant. And sometimes we have these just in particular varieties. And it's often due to incompatibility mechanisms where the plant will not let its own pollen actually, it might receive it on the stigma, but it won't actually allow it to create this pollen tube here, which we kind of see being formed here. It's really fascinating. when a pollen grain lands on the stigma, whether it's receptive to that pollen grain or not, it will create this pollen tube. It'll kind of send it out from the ovules, from the ovary, through the ovary, and up through that style to meet with the pollen grain. So it's really kind of fascinating. However, cross-pollination between two different cultivated varieties can be ineffective due to incompatibility. And kind of same deal, it's just due to factors inhibiting that pollen tube from actually forming. The angiosperms are by far the most successful group of plants. They make up the vast majority of terrestrial plants. And that is mostly because they have co-evolved with insects and other animals as well, but primarily insects. And it's actually the same way with the insects. The insects are the most successful group of animals in terms of diversity, and that's because they've co-evolved with the flowering plants. So generally, for cross-pollination, we have it's mostly done by insects or by wind, especially in grass family. But then there are also some other pollinating agents in many different plants, such as water in some cases, snails and slugs, birds, and even bats. Especially like the night blooming plants. Sometimes they are pollinated more by bats and moths rather than the pollinators we have during the day. Here's a cross section of a pistil of a flower following pollination. Again, we have that elongated pollen tube that carries the male gametes, the sex cells, to the ovary, where they then have that genetic recombination that forms a full set of chromosomes and creates the genetic material required for a new plant, or many new plants in many cases. Let's skip over a little bit here. But just note that in the gymnosperms, the cone-bearing plants, they're done by cones versus flowers, so it's slightly different. They evolved a little bit earlier than the flowering plants and were an important kind of stepping stone in the evolution of plants. We also have a phenomenon known as parthenocarpy, however you want to say it. That's the formation of fruit without the stimulation of pollination and fertilization. And this can be good for us in particular types of agricultural production because no seeds are produced. You've probably seen parthenocarpic cucumbers out there. They're seedless cucumbers that are just a result of this phenomenon. It can also be induced intentionally in some fruits by the application of some of these hormones that we began to talk about, and we'll talk about more today. The particular ones that cause parthenocarpy are gibberellins and auxins. However, not all seedless fruits are parthenocarpic, and in some cases, we have another phenomenon going on known as stenospermocarpy, where basically we have pollination and fertilization occurring, but when the fruit forms, the embryo inside of the ovaries and ovules is not actually completely formed. Therefore, we don't have any viable seed produced. That's stenospermocarpy. Those plants, of course, we have to propagate them vegetatively. And I just want to throw in an example of stenospermocarpy that we intentionally do to make a more marketable crop. I'm sure everyone's eaten watermelon with seeds and without seeds. And of course, it can be annoying because there's a lot of pulp there surrounding the seeds, but the seeds are large and in some cases can, they're not edible, or, well, I guess technically they are edible, but they're not fun to eat, right? They don't, you've heard tales of people swallowing a watermelon seed and and then it growing in their stomach and causing them to erupt a watermelon plant. Well, of course, that's not true. But nonetheless, seeds can be annoying, especially in watermelons. For seedless watermelons, they actually undergo kind of multiple generations. They will take a variety of watermelon and expose it to colchicine, which is a chemical it's a natural chemical that is obtained from the crocus plant. And I have no idea who figured this out, how they figured it out, but that's just one of these miracles of plant science. Someone working in a lab somewhere noticed that colchicine, when exposed to particular plants, induces tetraploidy. And whenever we talk about ploidy, what we're talking about really is pairs of chromosomes. So, of course, we are diploid organisms. We have two sets of chromosomes. And our sex organs, though, are haploid. They have one set of, set of chromosomes, but one pair of chromosomes, so that they can genetically recombine, and that's where we get that genetic crossover. So basically, to produce a seedless watermelon, they induce this tetraploidy in a plant. So they have kind of two different varieties, one that has this tetraploidy, one that is the regular diploid variety, and then they actually allow those to cross-pollinate. And then they take that seed, and that seed then becomes a seedless watermelon because it produces triploid plants that tend to be sterile because they have an extra set of chromosomes, which then causes them not to produce seeds. Of course, seedless watermelons do often have these kind of, you can kind of see in the picture here, little white infertile seeds that do not produce completely. It's kind of the same deal with the stenospermocarpi. The embryo just does not develop. So in flowers, we have kind of accessory tissues that are involved in fruit formation. However, botanically, the true fruit is just the enlarged, fertilized ovary. And then that fruit develops in this kind of coordinated maturation of all these tissues and organs working together in the formation of the fruit. In some cases, we have flower parts kind of fused into that fruit. Like if you look at a blueberry, for instance, you'll see the sepals of the flower are still connected to the tip of the blueberry. In many plants, only a small percentage of the flowers actually end up developing into fruits, especially in tree fruit crops, where there are tons of flowers, but the plant cannot possibly mature as many fruits as there are flowers. In some cases, the plant aborts those when the zygote fails to develop. In some cases, we will intentionally remove flowers and some fruits in order to develop larger fruits. Here's just a graph from the book looking at days from pollination to fertilization into the development of the fruit itself. And that can happen a variety of days. Some crops, we can have it rapidly, like cucumbers or many of the cucurbits, other squashes, beans, for instance. Many of these, they develop rapidly and over the course of just a few days. In some cases, they develop very slowly. Pineapples, for instance, we have growing here in our greenhouse, and it's interesting to watch. They kind of form this aggregate fruit, and each one of those cells kind of needs to be pollinated in order for it to develop. Of course, it is a self-pollinating plant, so it doesn't require any insect to do that. It does it on its own, but as those cells develop, the pineapple expands, but it takes quite a long time. It takes about a month for the flower to fully form, and then it takes about 6 months to go from fertilization to a ripe fruit. As you can imagine, at least that's in our greenhouse. I'm not sure if that's the same in tropical parts of the world, but here with temperate this temperate zone, with the amount of sunlight we have, it's a very slow process. Plant hormones, we're going to talk quite a bit about today, and they can be involved in fruit setting. However, the actual physiological mechanisms of that are still, to this day, unknown by plant scientists. And in some cases, we can actually externally apply these plant hormones to replace the stimulus of pollination or fertilization. And the particular application of other hormones can induce other fruits to set better. So one big thing in fruit production in agriculture is just obtaining the optimal level of fruit setting, and also the sizing of fruits to, kind of correspond with that. Of course, with too low of a fruit set, then we have lower yields, and that can result in an unprofitable crop. With too heavy of a set, we get undesirable, small, or poor quality fruits that tend to mature late. And to overcome excessive fruit set, often we will actually go in and remove flowers or fruits at an early stage in order to thin those out. And probably that record watermelon, that record pumpkin at the state fair, that was a result of this. They were removing the female flowers and any fruits that set in order for the plant to kind of use all of the energy from its photosynthesis to develop one fruit instead of several fruits. And it's not, there's probably some sort of mathematical equation there. I'm sure it's dependent on the species. But as far as I know, there's not any one rule in terms of, in order to get this size of apple, I need to, thin this many fruits. It's just kind of a result of experience, and the cumulative experience, I should say, and knowledge of many different growers. However, some fruits, the species are self-thinning. They will automatically drop a lot of fruits on their own. Again, this kind of all goes back and relates to the plant's goal of life, which is to reproduce and to create, to further its own genetics, basically, to continue developing new DNA. So we're going to look at some hormonal substances, and I'm just going to kind of highlight the ones I think are most important and most applicable to a lot of crop growth. So one big one here is the application of gibberellin to grape clusters at an early developmental stage of the fruit, which increases fruit size. And, again, some plant scientists out there was experimenting, saw this happen, and today this is a common commercial practice. Almost all the table grapes that we see, not just of this cultivar, the Thompson seedless, but really of almost all cultivars, are now treated with gibberellin because it causes the grapes to be larger. I'm sure you guys have probably seen maybe some heirloom varieties of grapes. Generally, the fruits tend to be round, right? They kind of tend to be spherical. But when you apply this gibberellin, it causes them to be more oblong. And I'd say probably most of the grapes that you guys consume are this oblong grape that is a result of this gibberellin application. So things we do in plant science can have market effects on the economics and the production of crops over long-term trends. So what happens during the flower development in the early stages of fruit growth? We have basically a period of intense cell division that causes most fruits to increase in size because of really not individual cells enlarging, but the entire, all of the cells within the fruit continuing to divide, and more cells are there. And there are kind of two basic patterns of growth for fruits. Either where it's a simple growth curve where it just kind of slowly begins to expand and at kind of an even rate of maturity. And then there's a double kind of curve where that's repeated, where basically there will be a delay. And really that's mostly due to physiological developments within the plant, where first it might just be the endocarp developing outside of the ovules, but then it might actually be a second phase of fruit development resulting with the maturation of the seed inside. So a couple of different things going on there. Some of the fruits out there will ripen with respiration and release this gas known as ethylene, which allows them to continue to ripen after they are harvested. And these are known as climacteric fruits. And these are all these fruits, apples, apricots, bananas, and tomatoes, release ethylene on their own. However, in many cases, we actually will harvest these fruits maybe close to ripeness or unripe, completely unripe, and then actually hasten the ripening either before or after harvest with ethylene gas exposure. So in some cases, we actually have in the transport of the fruits, or even when they reach their destination and before they're actually put on display, they're being gassed with ethylene, which itself is a plant hormone that causes the fruit to continue to ripen. And so many of our slicing tomatoes that we see in the grocery store, there is a reason why, in my opinion, they're awful, because they're harvested green. And you see that difference in flavor in vine ripened tomatoes, right? They also often taste a lot better. They're not gassed with ethylene because they're harvested when they're fully ripe. There's a lot of genes that kind of go into that. We talked a little bit about the genetically modified, or genetically engineered, I should say, tomato earlier on, the flavor saver tomato, where they introduced the genes. I'm not sure if this is true or not, but they say of a salmon to where basically it would be able to be transported better. It would be it could withstand cold more, to where it could be transported in basically cold trucks, and then it makes it there. It's still vine ripe and it should have better flavor, but really that whole flavor saver issue, the texture and flavor of the tomato was awful, and it was never a commercial success. Of course, the lifespans of flowering plants vary vastly. In some cases, we might have just a few months of a life cycle. In some cases, we have thousands of years. Actually, they believe that the oldest California coast redwood is actually closer to 4,000 years old. It's an exceptional individual that is almost, and actually probably now, over 400 feet tall, tallest plant on planet Earth. So again, and that's not just with wild plants. Like pictured here, they have this olive tree in the Mediterranean that was probably cultivated during the Roman Empire, so several thousand years old. Senescence is basically, we senesce too, but in plants, it's the terminal and irreversible deterioration of basically plant tissues, which then leads to cellular and tissue breakdown, eventually death. It's a very important part of plant life, because without senescence, we don't really get the full maturation of the plant and the end of its life cycle. All plants have a life cycle, even perennials. We might be able to continue that longer in some cases with some of our care, maybe some propagation, but they all have some life cycle, and it's not out there in the literature. It probably depends on each plant itself. If we were to grow a tropical fruit in a greenhouse, eventually it's going to die. It's going to just reach the end of its lifespan and die. So you can't just grow things in perpetuity. Or can you? There actually are maybe some ways to do that. Different life cycles, different plants and annuals. It happens in one season, as we know. And perennials, the tops of the plants die back, but then the shoot regrows from the crown, and the roots can live for many years. In deciduous woody perennials, the leaves senesce and die, but the shoot and root systems remain alive. And we do vegetatively propagate plants to extend their life in some ways, which theoretically, again, could be indefinitely, but in practice that doesn't really happen. It seems that eventually it will run out. Although there are examples out there, such as what they list here, the winter pearmain apple cultivated in England. They say it was grown there. They have records of it as early as 1200 BC. So we don't know. I mean, in some cases, we really don't know how long these plants could be vegetatively propagated before they begin to kind of abort themselves. It's probably different for perennials than it is for annuals, or at least what we grow as annuals. Same with the black Corinth grape, been grown in Greece for thousands of years. We took a trip one time to a guy that grew bonsai, and he actually, for a long period of time, maintained this bonsai collection in Washington, D.C. And he was fond of saying, oh, these plants are thousands of years old, when in reality he just vegetatively propagated from a plant that was thousands of years old. So he's extending the life of the plant by doing that. In some cases, though, some of those bonsai actually were, several thousand years old. Senescence isn't necessarily just caused by the genetic cues of the plant, but it can also be caused by pathogen attacks or environmental stresses as well. Plants decline, right? And that can trigger senescence if they're overly attacked. Senescence is not necessarily entirely just the degradation of the plant either. It's not just a bad thing. We have particular messenger RNAs in some proteins that are synthesized only in senescing tissues. Some fruits, for instance, really are only achieve their peak flavor, and actually, in some cases, their chemical constituents that could be required for medicinal purposes only when the plants are dying, they're senescing. The senescence is also hastened by the transfer of the stored nutrients to the reproductive parts of the plants. And it can be postponed in many plants by picking of the flowers or fruits before seeds start to form. And we intentionally do that in many cases. Again, we're kind of interrupting that plant's life goal of developing fertile seed and dispersing. So basically, think about, again, like these fruits that tend to develop very quickly, like squash, cucumbers, etc. If you allow them to develop and completely mature their fruit, that triggers senescence. But if you continually harvest the fruits, then it delays senescence. And so that's extremely important for crop production and being able to harvest our crops. All right, so we're going to get into plant growth regulators and hormones. And we do have to kind of distinguish the two. First, a hormone is the natural substance produced by the plant that triggers different activities and different forms of growth in the plants. Plant growth regulators, which we generally abbreviate as PGRs, often are plant hormones, or they are modeled after plant hormones. And so again, they can be natural or synthetic. And they can also be from chemicals that are not found in plants altogether. The plant growth regulators are basically kind of like hormone-like substances that are applied to plants in order to try to trigger artificially some of these different cues in plants in order to promote different agricultural activities. We got 5 traditionally recognized groups of natural plant hormones, and in this table we see the naturally occurring plant hormones and then also the synthetic versions of them. So the 5 categories are at least traditionally recognized categories, because there are more categories of these coming out all the time as the science further develops. But first we have auxins, we have gibberellins, cytokinins or cytokinins, ethylene, and abscisic acid. These are the ones that were kind of historically recognized, and we have naturally occurring forms of them in many plants. For instance, if we look at the auxin, this indole acetic acid is naturally found in willows, in the buds of willows. So if you take willow branches, for instance, and you put them in water, then they naturally will exude this indole acetic acid, which is this form of auxin that triggers rooting in plants. And it's really easy to start cutting some willow as a result of that indole acetic acid. You can also take that leachate that has been produced from this kind of tea, and apply it as a natural plant growth hormone or plant growth regulator in some cases. So below here we have the synthetic forms, and again you can see that they're modeled after the natural forms of the chemicals. In some cases slightly altered because they have to be. Some of the more recent hormones that have been added include racemolides, salicylic acid, and jasminates. So let's talk a little bit more about each of these. First, auxins, and they operate at cellular levels. And basically they originate in the meristems of plants, where we have these enlarging tissues and the dedifferentiated, or undifferentiated, I should say, plant cells. And they are, a result of or cause that apical dominance, which is basically the blockage of this lateral growth buds by the presence of this auxin in the terminal buds. Oh, sorry, one thing I want to point out there. Some important commercial uses of synthetic auxins include adventitious root initiation. Most of our rooting hormones are based on auxins. Weed control can also be accomplished through auxins. Inhibition of stem sprouting, and also tissue culture. Gibberellins are mostly through gibberellic acid, and they stimulate cell division, cell elongation, and can also control the production and secretion of particular enzymes in plants. So we use gibberellic acid as a way to, naturally in plants, it's the fruit set and growth, the maturation and the ripening in particular species, and in some cases can even control the sex of the plant. Last thing there, they were talking about gibberellins. They found out about gibberellins controlling growth of plants by finding gibberellic acid in dwarf plants. So here we have an untreated, genetically dwarf corn plant, and then in the center a non-dwarf corn plant that has been sprayed with gibberellin, and then on the right a genetic dwarf corn that has been sprayed with gibberellin. So basically, it was the one on the left, the dwarf plants were not producing gibberellic acid, which could be good or it could be bad in some cases. For production, it's a bad thing, right? Gibberellins are also involved in overcoming dormancy in both seeds and in buds. And it's kind of interesting. They were first discovered by Japanese researchers in 1926 by looking at a disease of rice plants that basically cause abnormal growth due to the presence of gibberellic acid in the fungus. And then here's what we were talking about earlier with the grapes. This is in the black Corinth grape. On the left, we have untreated. That second grape cluster there has been treated through girdling the stem, which basically causes an imbalance of the hormones. And the last two, different rates of gibberellin applied, and 20 parts per million. That last one gets kind of that full cluster of grapes, a little bit larger grapes. It can also affect the germination of plants too. So here we have gibberellic acid on grape seeds. As you can see, the higher the rate of application of gibberellic acid, the more germination we have, and also the more vigorous the seedlings are. A couple of things that we do with, and they say in the book here that there are only a few agricultural uses, but then they talk about quite a few different uses of gibberellic acid. Promoting male flowers in cucumbers, overcoming cold requirements for some plants, and in some ornamental plants causing different growth forms by causing stem elongation. And we got an example of that here. Something I want to point out real quick, we can actually induce some of this, not necessarily just by adding the hormone itself, but by pinching that apical meristem. And often it's not just the presence or absence of the hormone, but the amounts of hormones and the ratios of them in representation with other hormones. So here we have some poinsettias that are being grown kind of more in like a tree shape by using gibberellins in their vegetative form to basically elongate the stems. The next class are the cytokinins, and they promote cell division, but also do some growth and development, cellular enlargement, some tissue differentiation, can't speak today, as well as some dormancy. They basically do a little bit of everything. But they are often, we look at them as interacting with auxins to influence the differentiation of plant tissues. And it's the ratio of cytokinin to auxin that causes plants to put more of their energy in rooting or shoot development. And there are over 100 known natural and synthetic cytokinins out there. And here's just an example here of both cytokinin and auxin treatment on the growth and formation in tobacco stem segments used for, this is for tissue culture, which is kind of a plant propagation cloning technique where you can just take basically even just one cell of the plant and create a new plant, as long as you can use the right ratio of cytokinins and auxins to differentiate the tissues to develop shoots and roots. They also indirectly increase enzyme activities and increase the DNA produced in some tissues. Next, ethylene is a gas that, for a long time was not really known as a plant hormone, but today we really classify it as a hormone. It readily diffuses throughout the plant and moves much like a carbon dioxide or other gas. And it can exert its influence in very minute quantities. The synthetic ethylene form, there's a product known as ethifon, where we apply that to plant tissues to cause a natural burst of ethylene production. That's often what we use in the ripening of crops. So we have both ethylene itself, and then we also have ethylene releasing chemicals, which again cause fruit ripening, pre or post harvest, flower initiation, also flower fall. It can also change the sexual expression of flowers and organisms themselves. That degreening of oranges, lemons, and grapefruit is, again, it's kind of more ripening, really. Harvest age and growth. Ethylene, of course, can also harm plants because it causes unwanted leaf abscission or hastens the senescence of most flowers. And we've seen this. It's kind of funny, actually. We had a greenhouse manager here, and we had an adjunct teach a floral design course, and our greenhouse manager would often store her lunch in the floral cooler, which would cause the flowers in there to continue to basically produce and try to develop into seeds, which by the time the fall class rolled around, it was like, well, some of these flowers have kind of hastened their ripening and are overripe or dropping their petals now. So there can be unwanted effects of the ethylene, of course. And I would actually, if you guys want to conduct a little experiment at home, it's pretty cool to see. If you take an apple or even just parts of an apple and put it in a closed airtight container with, for instance, like a potato, it'll cause the potato to sprout. If you put it in with a flower, it will cause the flower to quickly senesce. And there are many different things that can actually give off ethylene. The storing of some fruits, along with other things such as bulbs or other fruits, other flowers, can cause negative effects. You can also have ethylene being produced in gas and oil-fired heaters that are not vented properly. So it's not just from plants. Then we have abscisic acid, kind of our last form of the naturally occurring plant hormones, at least traditionally recognized. And abscisic acid, of course, causes the abscission of leaves. It regulates processes in seed development and triggers dormancy in plants. And it is synthesized both in leaves and roots. Really, we don't use it often for commercial uses. So here's the 4 additional kind of classes of hormones. The brassicolides are steroids, and they're actually pretty similar to animal steroids such as estrogen and testosterone. And we're still figuring out how those could work and could be used in crop production. Salicylic acid is actually comes from the willows, and also from willows and some poplars too. It's the biologically active component of aspirin, and is an important component of plant response to pathogen attack. So again, we're still figuring out how to use salicylic acid to try to trigger plants to be more naturally reactive to pests and pathogens. Jasminates, also kind of the same deal. They are in the response of defense mechanisms of plants against plant feeders, herbivores. And finally, we have cysteamine, which is a unique plant hormone. It's the only one that is known to be a peptide, composed of amino acids. And it is naturally produced in wounded tissues of both herbivores as well as some plants too. So these are kind of newer. So we're still trying to figure out what we can do with them. Finally, we've got some synthetic growth retardants out there, and we use these to control forms of plants, often in ornamentals, to give them darker or more attractive foliage or kind of more dense growth. They usually block gibberellin acid. And the big one I wanted to point out here is paclobutrazol. We often use that in trying to keep chrysanthemums in vegetative states before they flower. All right, that is that for the second part of Chapter 7. Anybody have any questions, comments, or concerns? All right, well, you guys have a wonderful day, and take Chapter 6 quiz is up and will be available for a week, and then Chapter 7 I'll probably put up on Wednesday.
Okay, bye everybody.
Y'all have a good one.